Hello everyone, I am here Armaanti, and I would like to say many thanks as I and my team get this opportunity to present our research here in this international conference. And my team, they are Bapak Profesor Amran Laga, Ibu Dr. Andi Hasisa, Ibu Dr. Niken Darmayanti, Ibu Dr. Resmi Rumenta Siregar, and Ibu Atik Nurseha. I thank them for their support. And our research here entitled Screening Marine Lactic Acid Bacteria of Brown Seaweed, Sargassum pond system symbiotic producing bioactive compound from Indonesian waters. And to take benefits of the time, we will move on to the main presentation. Well, this is the presentation. As introduction here, we are talking about primary and secondary metabolites. So we understand that the potential of Indonesian marine resources include fish and non-fish biological resources. And seaweed here is a very important biological resource. They are the largest producer of biomass in the marine environment. And we're also talking about Symbian bacteria because we also understand that this bacteria can live in everywhere. Microbes also can present as a living symbiont in association with various algae in the form of epiphytes or endophytes. And talking about antibacterial, brown algae and other species have been extensively analyzed for their antibacterial and antifungal activity. But here we're not talking about the antibacterial from the brown algae. Instead of, we're talking about the we want to see the Symbian bacteria ability in producing antibacterials, because some research already mentioned that this Symbian bacteria from marine environment may have better potential in inhibit the growth of pathogenic bacteria. Well, that's the introduction, and we will see the materials. And here we have equipments and materials. Equipments that we use based on the laboratory standard equipment, for example, like petri dish, reaction tubes, until microscopes. And for the materials, materials were used conducted this research include brown algae, that is Sargassum pond system. We also need pure culture bacteria of Staphylococcus aureus and E. coli. We also need some medium for growing the bacteria, and also some medium for cell staining. Okay, for the method, we have here 4 main procedures. The first procedure is a sampling. So the location of sample derived was in Lima Island. It was in Banten. And sample collected about 100 grams. Sample maintained in fresh seawater with oxygen addition for further laboratory analysis in less than 24 hours. And the second procedure here is a bacteria isolation. So isolation was started by enrichment sample from the algae. We did this by inserted into a 30 milli MHB medium. And we shake the sample at room temperature for 24 hours, and then sample diluted with 9 milli MHB until 5 dilution. So each this dilution was grown on an MHA medium, then incubated in a specific time and also temperatures. And here the colonies of bacteria that produce antimicrobial are characterized by a clear zone. For algal diffusion test, we prepare paper dish containing 40 microliter of supernatant, and we also need negative control, and also we need positive control of chloramphenicol. And they were placed on the surface of MHA containing 1 milli of the test bacteria, and they incubated and observed the zone of inhibition. The last procedure is a bacteria identification. And here the isolate was observed morphologically. Also, we did the DNA test to know the species of this bacteria. Okay, that's the method. And here we have result and discussion. And as a first result, we have bacteria isolation result. So we identify this one colony, and the chosen colony show a round shape, white color, flat edges, and has convex, shiny elevation. We have here the second result. It is paper disc diffusion test. An application of antibacterial compound produced by these Symbian bacteria show different inhibitory activities against the two bacteria tested. As we can see in the pictures, that paper disc with the supernatan applied to the plates containing Staphylococcus aureus show a stable clear zone even after an incubation period reached 48 hours. So we can say that this supernatan work better against the Staphylococcus aureus as a gram-positive pathogenic bacteria. It is inhibition zone diameter result for a gram-positive and the gram-negative. And Symbian bacteria can produce around 7 millimeters zone of inhibition against this gram-positive bacteria. And for gram-negative, the negative control and even the Symbian bacteria didn't produce any zone inhibition, and the positive control, chloramphenicol, also work better against this gram-negative. So here we have the third result. It is identification of Symbian bacteria. But we understand that this bacteria has rod shape. It was not acid-fast, not forming spores, and it is not motile. There are aerobic growth and negative catalase and also positive carbohydrate test. And after testing the DNA, it is known that the species homology of the tested isolate was Lactobacillus plantarum. And as a conclusion here, finally, we can understand that this lactic acid bacteria marine strain may open up opportunities for their utilization, especially in food processing or other purposes. Okay, well, I think this is the end of my presentation, and thank you for your kind attention.